Well, welcome everyone to our second XLA Life Fireside Chat. We are honored to have Dr. David Rawlings as our guest speaker, our very first guest speaker. So before we get to the gene therapy discussion, we're going to go through a few things with XLA Life. So here is our agenda. We're going to do XLA Life updates, and I'm going to ask a few polls to the participants. Then I will introduce Dr. David Rawlings, and then we'll have our presentation on gene therapy for XLA, followed with a questions and answers session. And for that, I will ask that questions, you click the Q&A button. The chat is for everyone to see. That's for discussion. So you can chat with anyone who's participating in today's webinar. And we will address the Q&A answers that we get live in the chat at the end. So to start, we have some updates from XLA Life. We just last week, we got our approval for 501c3, so we are tax exempt. So if you ever need a place to donate money, please visit our website. We'll have a QR code at the end for donations. We're working on our next XLA Life Fireside Chat, date to be determined. It's probably going to be mid to late July. So be on the lookout for updates there. And finally, just a couple weeks ago, we were introduced to the Primary Immune Deficiency Treatment Consortium, the PIDTC. This is a collaboration of over 44 centers across the globe. The best in immunology research and patient advocacy all participate there. They have several active protocols on functional treatments for severe combined immune deficiency, Wiskott-Aldrich, CGD, and primary immune dysregulation disorders. However, there's little to no XLA representation so far, so I'm hoping to change that. I'll be a fly on the wall for the next few meetings, get a better idea of how we can help advance XLA research there and how we can be a benefit to the PIDTC as well. So that is it for XLA Life updates. And now we're going to start off with some polls. We want to hear from you, the XLA Life community. And our first question is, how do you feel about gene therapy? So 1, being very hesitant, and 5, being very optimistic. Feel free to answer however you like. We will also be asking this same question at the end, and so we can compare how you feel at the beginning of the webinar versus the end. So we'll give another 10 seconds or so to get some responses in and poll, and here are our results. So fairly optimistic. That's cool to see. Yeah, so we'll see if we can get a few more optimistic folk at the end here. And so we have one more question, which is, how informed do you feel on the concept of gene therapy? So 1, very uninformed, 5, well-informed. And this is really the question we hope that today's presentation will really see a difference between the first ask at the beginning of the presentation and at the end. So we'll give this another 5 seconds or so. We're up to 14 responses, which was just one less than before. All right, and poll and share the results. So the plurality goes to kind of uninformed at a 2. So we're hoping to get that number up, and I'm sure Dr. David Rawlings' presentation will do that. Okay, and so on to the introduction of our guest speaker today, Dr. David Rawlings. So Dr. David Rawlings is a professor of pediatrics and immunology at the University of Washington. He directs the Center for Immunity and Immunotherapies, the CIIT at Seattle Children's Research Institute, and is the chief of the Division of Immunology at Seattle Children's Hospital. Seattle Children's is the major referral center for pediatric and adult patients in the Pacific Northwest and nationally with genetic immune disorders. So Dr. Rawlings leads the basic and translational immunology research programs over there, including over 14 independent investigators focused on human immune disorders. He also leads the program for cell and gene therapy, developing gene replacement and gene editing approaches for genetic immune diseases, just like XLA. His independent research group includes approximately 25 members focused on studies of altered lymphoid development that lead to immunodeficiency or autoimmunity. But his main focus is on gene therapy. And these kinds of accolades are very hard to come by in any field. So we are incredibly lucky to have Dr. Rawlings focused on XLA. I first saw him speak about XLA gene therapy at an NIH webinar back in March. I knew I had to reach out, and shortly after, we were on a video call talking about everything about XLA and even some rock climbing. We're incredibly honored to have him as our first Fireside Chat guest speaker. And so without further ado, I will hand it over to Dr. Rawlings.
Thanks, Austin. Can you guys hear?
Yeah.
I'm going to share my screen here. So it's really a pleasure to share our work with you guys today, and no pressure on getting the poll to change, I guess, but I never had that at the start of a seminar before, so that's extra pressure. Hopefully I'll do a good job and try to help explain the concepts about gene therapy, and then in particular what we're trying to do to make a gene therapy specifically for XLA, and the steps that we've taken and where we are and where we're going. So I'd like to start with this slide, which is kind of gives you a little bit of historical background on me and my interest in XLA. When I was a fellow at UCLA, we discovered BDK, and we discovered its role in XLA, and then also in a mouse disease that was similar to XLA called XID. And so I've been working on this actually since before 1993, because we had to do a little work before we actually discovered this. So it's been quite a while, since the early 90s. And since that time, at that time, I had already was interested in gene therapy and became very interested in gene therapy for XLA. And it's taken quite a while to get where we are today. And you'll see kind of all the steps that are required and why it's taken some time. But we early on made a knockout of BDK in mice, and then we made an even better model that we call mouse XLA, so we could study treatment for XLA in mice. And we showed early on that an early stage of a viral vector called a retroviral vector could rescue XLA. But in the last decade, we've worked on making a more definitive and better therapy, which I'm going to tell you about. And luckily, we're able to partner recently in 2020 with CSL Behring, who many of you probably know, and some of the CSL Behring folks are on the call today. And we're now at the cusp of preparing the investigational new drug application for the FDA. We've had discussions with the FDA, and we're on the cusp of actually setting up this clinical trial. So really interested to hear your feedback and questions about where we're going. So just a quick introduction. It takes a village to do what we're trying to do here. We have clinician scientists who are leading our clinical team, Alex Petrovic in particular, who was unable to join today because she had a family emergency, but she's going to be the site PI for this clinical trial. She's trained as both an immunologist and a bone marrow transplanter, which is critical to kind of the expertise needed for this trial. Our own preclinical team in our lab involves many different people. Some are on the call today. I'll point out in particular Karen Summer, who's worked in my lab for almost 20 years and did some of the vector development that I'll be talking about. And then new members of the lab who are really leading this effort. Danielle, I think, is on the call also, and Laura, who's helping us coordinate all of these efforts, and Amalia, I think, is also on the call today and is working on our human cell therapy translation of our work. And then I'll just point out another large team that we work with, which we call the Cell Manufacturing Group, or the Therapeutic Cell Production Core. And this is an essential process that we call that is referred to as good manufacturing. So in order to make a cell product to give to patients, you have to do this in a very controlled and precise and safe way. And so our TCPC group has lots of experience. They've made many different cell therapy products. They have at least 10 active trials right now. And we're excited. In the last year, we opened at the top of this beautiful building in this picture this building's called Building Cure. And at the top floor of this building, we have the largest GMP facility in the pediatric hospital in the US. And so lots of good expertise to make the cells that we're gonna use in a cell therapy trial. So why do we do a cell therapy for an immune disease? And this is really the key question and concept. So all of us in our bone marrow have this rare and special cell that we call a hematopoietic stem cell, or hematopoietic stem cell, or HSC for short. And these cells can turn into all the different types of blood cells in the blood. So they can turn into lymphocytes. They can generate what are called the myeloid cells, including the neutrophils that fight bacteria, for example, and also the platelets that help stop bleeding, and our red blood cells. So this one type of cell gives rise to all of these cells. So the idea is that if you can make a genetic change in a hematopoietic stem cell and then give it back to a person, then you can generate the cells you're interested in fixing, that might be missing or not functioning normally in a disorder. And in the case of XLA, the biggest deficit is in, as you guys know, is in B lymphocytes and their development. But also, there are also some functional deficits in the myeloid cells. And these are the two types of the B cell, and the myeloid cells are where BTK is expressed. So in order to have a therapy that is optimal, we want to express the normal BTK gene in cells which become B cells, and also cells that become myeloid cells. So how do we, so what does BTK do? Just to get us all sort of on a kind of a starting page on understanding BTK. So BTK is most involved in B cells, as I just said, and B cells have on their cell surface a protein that's here shaped like a Y, and that protein is called the B cell receptor. And it's really critical for B cells sensing pieces of bacteria or viruses. So when B cells can see these infectious components, it triggers the B cell receptor to start signaling. And when that signaling begins, BTK sits in this group of enzymes that are in the cell, and BTK gets activated through complex things, which I won't tell you about. And then following the activation of BTK, there's all these biochemical events that happen. And the take-home message is these biochemical events are really critical for the development of B cells, for their division and growing, and also many other functions of B cells. So when BTK is absent or not functioning normally, many of these signals don't work, and then we have the developmental problems that happen with BTK. And in that long timeline that I showed you at the beginning, one of the first things we needed to understand was what BTK actually did. And so we spent a lot of time defining these biochemical pathways in cells, and we now have a really good understanding of what BTK does, and that helps us understand how we want to make it work properly. So if we think about B cell development, which is shown here in this schematic, the hematopoietic stem cell generates a cell that we call the pro-B cell, and these cells go through a series of developmental steps to become mature B cells. And it's the mature B cell that can get activated and make a plasma cell that makes antibodies, or turn in what's called a memory cell that can come back and remember an infection and then make antibodies later. But in order to have this happening, you have to generate these mature B cells. And BTK is required at multiple steps to generate these immature B cells, and then also to allow the mature B cell to function normally. So so normally in the bone marrow, our pro-B cells divide, and they make this cell called the pre-B cell. And this pre-B cell usually undergoes a big division to make a lot of pre-B cells. And that process in XLA is blocked. So in the absence of BTK, this expansion that happens and then the generation of those mature B cells is blocked. And that's the big problem with B cell development in BTK deficiency or XLA. The other key thing that allows us to think about is that if we are able to provide a copy of the BTK gene into the stem cell and then the differentiating cells, we know that these cells have an advantage over the cells which are blocked. And so this selective advantage allows gene therapy to potentially work really well in patients, in people with XLA, because we think that even fixing a small proportion of the hematopoietic stem cells will allow this selective advantage to happen. And I'll show you that is the case when we study this with our gene therapy approach. So how do we do gene therapy and what are the steps? So it's really a 3-step process. We have to collect the HSCs from the person with XLA. And this is actually quite a fairly easy process now. And we can talk about that in the question and answer session about how that's done. But when we get these stem cells, then in that cell manufacturing laboratory, the GMP setting, we would have to then deliver a corrected copy of BTK into these stem cells. Then take those corrected cells and give them back to the same person that donated them. And so this is what we call it's the equivalent of a bone marrow transplant, but in this case, it's a self bone marrow transplant where we've done something to the bone marrow stem cells to make them more functional. The other point that's important to understand here is, while this is a self transplant, we still need to make some space in the bone marrow for these cells that are coming back into the bone marrow environment. And that requires something that we term on this slide conditioning, but what it really is some kind of dose of chemotherapy that makes some space in the bone marrow for these cells to go in and go back and live in the bone marrow again and compete into the bone marrow. So there's sort of really, when we were developing this therapy, there were 2 essential questions that are central to a successful gene therapy. One is, how do we get the corrective gene into the stem cell? And the other is, how do we make sure that BTK is being made in the right amount and in the right cell types? And so I'm going to show you a little bit about how we got there, and we can answer more questions about that at later as well. So the first question is, how do you get a corrective gene fragment into a stem cell? How can you do that? And the answer is actually taking advantage of viruses. So as many of us know, viruses evolve to enter cells, and they carry in DNA and RNA into cells. So what biologists have done over the last 20 or so years is figured out how to utilize certain kinds of viruses. In this case, we're using lentiviruses. And what we do is we make a genetically engineered virus that lacks almost all of the components of the normal virus, but we can introduce into that virus pieces of DNA that we want to deliver into a stem cell. So these viruses can enter and deliver this DNA into the hematopoietic stem cell. They can't replicate their defect. We've made them defective, so they can't work like a normal virus. And they're well regulated and safe. And this has been shown now to be used successfully in a number of gene therapy trials for immune deficiency diseases, some of which are listed here. So having this platform of a lentiviral vector allows us to get a piece of DNA into a cell. And how does that work? So what does that look like? So we have this virus that has, in this case, a fragment of the BTK gene fragment. And this virus naturally can enter the hematopoietic cell in a process that we call transduction. And the DNA then gets released from the viral particle and is able to make its way into the nucleus. And the remarkable thing about these lentiviral vectors is that this piece of DNA can then get incorporated into the chromosome of that cell. So it becomes part of the DNA that will always be in that cell. And when the cell divides, that copy of the BTK gene will be copied into the next cell, the progeny of that initial cell. So when that hematopoietic stem cell divides, every cell that is a daughter cell of that hematopoietic stem cell will carry a copy of that gene. And so when that gene is present and it's regulated properly, then it expresses the protein BTK, and that protein then is present in the cell and able to do those signaling events that we talked about at the beginning. So then the question becomes, well, we have this way to package the piece of DNA and get it into the hematopoietic stem cell efficiently. And we know this works, but how do we assure that BTK is produced in the right amount and in the right type of cell? And that is really what this 10-year timeline was spent figuring out. So I won't bore you guys with all the different steps that we went through to figure out the best way to do this and the best type of lentiviral vector. But I'm going to show you kind of the end product of that, and then some of the results that we have with that, what we think is a really good clinical vector. So this is a complicated slide, and I'm going to use it just to make a few simple points. The most important one is that when we make this lentiviral vector, when it gets incorporated into the chromosome, it looks like this. And so it has this copy of the BTK gene that we're delivering. So this is the BTK gene, and it'll be the full length BTK sequence, normal BTK, unmutated BTK sequence. And then the most important part is this, which we call the BTK promoter. So this is a piece of DNA that comes from the human genome that is the normal piece of DNA that controls expression of BTK in B cells and myeloid cells. So we want to use, this is the natural promoter. So we're using the natural promoter to turn on BTK in the cells where it normally would be made. And this has allowed us to drive expression just in B cells and in myeloid cells. And the other key part of this vector is this thing that we call the UCO, shown in blue here. And this is another piece of DNA from a different part of a different chromosome. And it has a special property where it keeps the DNA around it open so that it allows this promoter to stay active in all of the cells where this piece of DNA has become integrated into the genome. And so we found that using this piece of DNA remarkably enhances the ability of this promoter to stay active in the cells that we're interested in. So this is a key. These two pieces are really critical for this vector to work. There's a couple other things in here which are not that important to understand, but this thing called the WPRE and the poly A signal are critical for the transcript to be processed properly and expressed at good levels in cells. So that's the vector. So does it work? And so we have set up this model that we call the XLA mouse model. And what we essentially do to test this is we harvest the stem cells from XLA mice. We use our XLA lentiviral vector to introduce the gene into the genome. and then we transplant these cells back into other XLA mice. And we do this in a way that we can distinguish the cells that came from this donor mouse from the cells that were in this host mouse, so that we can track what happens. That's why they're shown in white and black here, just to kind of see the difference. And so we do a very extensive analysis, and what we are looking for is, did we rescue XLA? Do we get B cell development in the right types of B cells and the right numbers? Do we see BTK expression in the cells we expect? Do the B cells function? How many copies of the virus are there? And then we do all these other experiments to look at the long-term health of those animals to make sure that this is safe. And we do this in large numbers of animals, and we've studied this extensively and published work on it. But I'm just going to show you, and I don't want to show you a whole lot of data, but I'm just going to show you a tiny bit of data here. Just so each of these circles is a mouse that got treated. The black circles are mice where we just transplanted normal bone marrow back into the XLA mouse, so that you can see in this case, they get lots of B cells in their bone marrow. In contrast, if we take the XLA bone marrow and put it back into the XLA mouse, there aren't any B cells. But if we use gene therapy treated stem cells, you can see in the bone marrow, about half of the cells are expressing BTK. But then when we look in the spleen, which is the organ that has the most of the B cells in the outside of the bone marrow, and is similar to lymph nodes, but it's really like a gigantic lymph node is the way you could think about the spleen. And in this case, you can see that the gene therapy treated animals have almost exactly the same proportion of BTK positive cells in the spleen. And this is a way that we do a cell count of the different types of B cells in the spleen. You can see that in the wild type and the rescued animal, we get in the wild type, in the control animal and the lentiviral treated animal, we get normal numbers of B cells. And there's lots of other data. I'm not going to show you it, but we show that this vector can restore BTK expression and development and B cell function. And I think it's important to kind of highlight this idea. So on that slide where I showed you in the bone marrow, we saw maybe we see somewhere between 30 and 50% of the cell of the developing B cells expressing BTK, but in the spleen and in the periphery, see almost all of them. And so that's a direct demonstration of this selective advantage that I talked about, where you can partially correct the stem cells and get a selective advantage to correct the number of B cells throughout the body. So this work is published, and I encourage you guys to look at it if you're interested to look at it in real detail. But we showed that this expression not only is in B cells, but it's also in those myeloid cells where we want BTK to be expressed. We rescued all of the types of B cells which are missing, and I didn't go through them one by one with you all, but there are many different types of B cells which are important. I also didn't show you, but obviously very important, we rescued antibody responses to vaccines. And then we also showed that that blue chunk, that UCO piece, was able to prevent silencing of the viral genome, which is really important because we want this gene to be expressed for the lifetime of an individual who gets gene therapy. And so having this element showed some real benefit. And we also, really importantly, saw no evidence for the vector creating any detrimental effects to these animals. So will this work in XLA volunteer hematopoietic stem cells? So we've had 4 adult XLA volunteers who came to our group and went through this process called apheresis to get stem cells. We isolated their hematopoietic stem cells in our GMP facility, and then we studied those stem cells for work in a model where we can take the stem cells and put them into an immune deficient mouse where human cells can survive, and we can test whether human hematopoietic cells, the biology of hematopoietic cells. So in this case, we take stem cells from our control volunteers or our XLA volunteers. We culture them like we would be doing in our GMP manufacturing setting, and then we deliver the XLA lentivirus or not, or we don't, and then we transplant those into these immune deficient mice, and then we look at those animals after several months. And what you can see is that in animals that received cells from the control volunteers, we see lots of B cells, but there are many fewer B cells in the recipient animals that got XLA stem cells. But when we treat with the XLA lentivirus, we get almost completely normal numbers of B cells, and the majority of these cells are mature. This model has allowed us to really test our vector in stem cells from patients, from our volunteers with XLA, and it showed two things. It showed that this animal system mimics the defects that we see in XLA. So B cell development stops in the bone marrow, like I showed you in our cartoon early on. There's nearly no mature cells in the spleen or in the peripheral blood. And the only cells that we find actually in the spleen are pre-B cells, the ones where they can't get past that developmental stage, but they do make it out into the spleen, so we can find them in the spleen. And then when we use the viral vector, we see this increase in B cell development, in significant increases in the total B cell numbers, and then we also can do some studies to show that the B cell receptor is working. So where do we go from here? And so this is, we've taken this lentiviral vector system and partnered now with CSL Behring, and we're going to make this product that we call CSL215. And this will be our first in-human phase 1/2 trial for subjects with XLA. And so I'll walk you through what would happen in a clinical trial. So we will have subjects that we do the process that we call apheresis. So this is a process where we give a medication that helps move the stem cells into the bloodstream. And when we use that medication, we can then collect blood from the individual, and there'll be lots of those stem cells in the bloodstream. And then we can take that product, that mobile apheresis product, and we can, in our GMP facility, isolate the CD34 cells, which are the hematopoietic stem cells. And then then we utilize those cells to then do that lentiviral delivery, and we then freeze those cells in a way that they survive. And then we do lots of testing to make sure that we got the viral vector in, and that the product is sterile and safe, and many other different what we call release testing. And only when that product is proven to be safe and meet our criteria, then our subject will begin what we call a conditioning protocol, where we give a low to medium dose of a chemotherapy agent called busulfan. And we are going to do this with a very tight measurement of the levels of this medication in the bloodstream. So we reach what we call a targeted level of the drug that we achieve very consistent conditioning. And then after that process, after those treatments with busulfan, then we would infuse this, we'd thaw this cell product and infuse it into the bloodstream. And so this would take place in the hospital, and then we would monitor our patient for a period of time to make sure that their bone marrow recovers from the conditioning process, and then we do a long term follow-up study measuring different aspects of the reconstitution and the function of those treated hematopoietic cells. So that's the big picture of how this is done. And initially, we will treat adult subjects with XLA because, as you guys know, XLA is not normally being treated with bone marrow transplant steps, although it has been done in a growing number of patients, and it has been done successfully with non-self transplants. But we want to demonstrate safety and benefit to adult subjects first. Obviously, the subjects will have to have a genetic and demonstration that they have XLA. And then we have a series of criteria that we will utilize for who would be eligible for the trial, which we can talk a little bit about in the question and answer if you have questions about that. But that's where we are. We're close to opening this trial. We don't have a precise date of opening the trial, but it's in the relatively near future. And so I'm going to stop there. Hopefully I haven't taken up too much time. And thank you for your attention, and really excited to answer any questions you have.
Well, great presentation, Dr. Rawlings. I have to say I'm incredibly more excited about gene therapy now. And so if you have any questions, please drop them in the Q&A box. We already have some questions off of Facebook, a few of you asked. And so I'll go ahead and start asking those. So you mentioned the near future, but I think everyone is itching to ask just about like what is the time frame for near future? Is it a couple of years? Is it a couple of months? I think a lot of people would be interested to hear that.
Yeah, it's most all these things lots of things have to kind of come together to be ready to go. But it's about a year to a year and a half is sort of the timeline that we're estimating, probably closer to the year and a half timeline.
That's great. So the first in-human you mentioned will be in adults 18 to 45. But what milestones need to be reached before this therapy is available to the wider XLA community, including those younger than 18?
Yeah. So that's the super good question. And we've had discussions with the FDA already. We've started having like a discussion with the FDA about the design of this trial and who would be eligible. And what we're proposing is to, and we haven't aligned entirely on this yet with the FDA, but we're proposing to treat a small number of adult subjects first, follow them for a period of time that we think will demonstrate safety and benefit. And then we will ask them to be able to open the trial to adolescent patients as the next step. And then we hope to treat a group of adolescent patients. And then with that data, we're hoping that the subsequent trial that we would run would include children. And that's our plan. Obviously, it depends on benefit and safety. But we're talking probably, from when we open the trial to when we could get to children, another five or so years probably in conservative figures. So it'll be a bit of time before we get to children. I think my view, and I think a lot of families would agree with this, is that that we do want to be able to treat subjects earlier in life because of the ongoing infections which are causing tissue damage to the lungs and sinuses, and also the other complications that people develop with XLA over time. And we really think that the greatest benefit may be in those in individuals who don't have damage already to the lungs, for example. Although I do think that even in subjects that have had some detrimental effects in their lungs will still benefit from this type of a therapy.
Yeah, absolutely. That's a good question. Those who already have some tissue damage, it'll be really interesting to see how gene therapy may reverse, if at all. So now another kind of candidate or eligibility question, and a little bit broader, but is gene therapy a viable option for any type of BTK mutation?
Yeah, so the way we designed this approach is that we should be able to cover any kind of a problem in the either the coding, like the gene sequence itself, or in the elements that control the turn up, like the expression of the gene or the downstream regulation. So essentially any mutation should be treatable with this kind of an approach. And that was why we designed it this way. Some subjects with XLA will have expression of a BTK protein, but it might have a mutation in the sequence, but that mutation that leads to a lower functionality of the protein. We know that we think that those patients should be equally treatable with this type of an approach.
And so while we're still on the approach, Steve has asked this question. How does Dr. Rawlings and his group approach compared to Dr. Cohn and his group at UCLA?
Yeah, so I think you're referring to gene editing as an approach, and we also are studying gene editing, and it's a different approach. So to sort of describe it in a way that everyone can understand is that when we use lentiviral gene therapy, we're putting the piece of the sequence of DNA gets inserted randomly in different places in different chromosomes. And so, but that's a very efficient process. And so we can efficiently deliver that sequence to the stem cells, and it's been shown to work really well in other immune disorders with controlling the number of viral copies of the viral genome that we introduce. In gene editing, what we do is try to edit the BTK gene itself in the stem cell, and in that case, we use an enzyme to cut the genome right at the BTK gene, and then we deliver a DNA fragment that rescues the BTK protein or sequence, very similar to the DNA fragment that we would use in the virus. So that approach is it's not as efficient as the lentivirus, but the biggest problem right now in the field doing that is that when we do that gene editing in the way that we have to do it to get the large piece of DNA into the genome, those stem cells are not as competitive as normal stem cells. And so they don't engraft as well in the models where we study it. And so we and many others in the world, across the world, are trying to optimize gene editing in a way that the stem cells are more healthy and work better. And if we can achieve that, then gene editing may be as good as, or perhaps better than viral gene therapy. But I think that we're a lot farther away right now from gene editing. And frankly, if viral gene therapy, testing viral gene therapy still helps us understand in the future whether gene editing will also be beneficial. So it may be that what we do now, for the next decade plus will be viral gene therapy, but then we may move to gene editing for XLA and other immune deficiency diseases. I think that we're all very interested in trying to do that, but it's further away from clinical application at the moment.
That's
a long answer, but I think it's important for you guys to understand where these two things sit in the clinical pipeline.
No, definitely. It's really important to understand the timeline of both approaches. And so while we're talking about the approaches as well, how does this current gene therapy approach mirror that used in SCID, so severe combined immune deficiency?
Yeah, so very similar, but there's some important differences. In SCID, so far, most of the SCID gene therapies haven't utilized the natural promoter sequences of the gene. They've used other promoters where we express the missing gene in all cell types. That we don't think is a good approach for BTK because we don't want to overexpress BTK in cells where it's not normally expressed. In SCID, many of the genes that we're talking about are expressed in many different cell types, so it's not, you don't have to have that regulation. But there are many immune deficiencies where the regulation is really critical, and we don't want the missing gene to get introduced into cells which normally wouldn't express it. And so that's a key difference.
So that's a good segue, talking about the BTK overexpression, making sure it's expressed in the right cells and at the right amount. And that can bring us into the potential pitfalls or risks that people may have. So Marnie has asked, this is a bit different, but does the, actually I'll ask you this one. Do you feel that there is a cancer risk from this gene therapy process? And how could that be treated or prevented?
So that's a really important question. And there are a couple points about this. One is that there have been cancers that have developed in patients treated with gene therapy with other viral vectors, the earlier versions of viral vectors before the lentiviral vectors. And those, we understand a lot about what happens there, and the lentiviral vector system has overcome most of those risks. However, we never entirely know the safety of anything. Unfortunately, we do all that we can in advance to test things for safety, but we don't know that, for example, BTK overexpression might be problematic in certain cell types. There is evidence that BTK is involved in certain types of cancer, for example. But we've thought about this a lot, and I didn't go into this, but when we designed this vector, we designed it in a way that it actually doesn't overexpress BTK. We have it actually expressing BTK slightly less than what we see in the normal cell. And so we did that on purpose because we knew that we thought we would get the selective advantage and the function of BTK without overexpressing it. And so we did that on purpose. We made that choice on purpose to make this potentially safer. Then the other comment I'll make is that whenever we use a conditioning step in a self-transplant or any other kind of bone marrow transplant, the drugs that we currently use for conditioning have some risk of damaging the stem cells of the individual that's getting that treatment. Now, that risk is a lot lower when the dose of the chemotherapy is low. And so we've chosen a low dose of this chemotherapy agent, busulfan, which is being used in essentially all of the other immune deficiency trials at different doses. And this is a dose that's similar to what's been used. And so far we haven't observed any evidence of malignancies that have come from the conditioning. But the follow-up time, everyone should realize the follow-up time for that is still relatively short. But we do have a long history of using busulfan in patients with other diseases, in particular like leukemia and other things where we've transplanted many children over a long period of time. And the secondary malignancy risk from busulfan is very low.
That's good to hear, because there are inherent risks with an approach like this, but it looks like you're taking every precaution necessary to really lower those, and even taking advantage of the selective advantage that BTK B cells have is a really cool trick to help that out. So that answered also one of Marnie's other questions. Well, she specifically asked, does the chemotherapy used affect fertility? But it sounds like we're at a low enough dose that we're lowering the risks of that
Yeah,
as
and
well.
we selected, and I wish Alex was on the phone here, but we very carefully thought about that and selected the dose of busulfan based upon demonstration that that did not impact fertility. But when we enroll subjects, we will bank sperm for every subject who's interested in doing that, and we have ways of doing that even in our very young adolescent.
Wow.
As well. So that's being done now in transplants, for adolescents at our institution to try to preserve fertility in conditioning settings, which are more robust, actually.
That's very interesting. I didn't even think of that part of the transplanting process. Now that might be a worry. So good question and great answer. And Terry has asked, so as well as capturing data on the increased number of B cells, what steps are you taking to see improvements in the myeloid cell types?
Yeah, so that's a really important question. And we haven't spent as much time studying this. We will definitely build in studies to look at the function of myeloid cells in the clinical trial. And we have studied the role of BTK in myeloid cells in our group, and there's complicated roles. And it's complicated because there's many different cell receptors that utilize BTK, but they don't typically, none of them in myeloid cells utilize BTK independently of other. There's a family of BTK-like proteins, and there's what we call redundancy in function of these proteins, which is why we think the myeloid function isn't impacted as much in BTK deficiency, because there's other family members that are similar to BTK that are serving some of those functions. But there are still evidence of certain kinds of infections, which are, where neutrophil biology, for example, is impacted in the absence of BTK. And so we're going to be looking for those kinds of outcomes.
That's really cool. When I was studying the BTK pathway at the NIH, I was amazed at how many roles we know it to play in myeloid cells, and the amount of roles we don't know it might play as well. It's a very tricky protein in those cells.
And I will say, we were eager to launch a clinical trial earlier for BTK, and we had a B cell specific promoter system that worked really well. But I decided at that time that I wasn't satisfied with that because of the expression of BTK in myeloid cells and its sort of growing recognition of its roles. So we kind of put that on hold and redesigned our vector system so that we would be able to get expression in myeloid cells for that reason.
That's great. So we only have 4 minutes left. I'd love to ask more questions and answers, but I do want to hear from the XLA community after the great presentation by the great Dr. David Rawlings. Could you hit next slide, please? And I will, in the meantime, I'm going to ask the same questions again on the poll, so please feel free to answer. How do you feel about gene therapy now? All right, 3, 2, 1. And this is much better than at the beginning. So we had a couple of very hesitant when I first asked. Now it looks like we've shifted up to the very optimistic, and I have to say myself, I am much more optimistic after hearing this talk as well. And so I will ask the fourth and final poll question here. How informed do you feel on the concept of gene therapy after the talk? And David, can you move forward in the slide deck?
Yes.
A slide or two, thanks. All right, 5, 4, 3, 2, 1. And this is great to see. So we had a plurality at the 2, kind of informed before, I mean, kind of uninformed. And now we see the plurality is at well informed. That is amazing to see. That makes me very happy, and we need to have more of these. That just goes to show it is really helpful for the community. So that is it for our second Fireside Chat. Please donate and visit at www.xla.life. Join our initiatives, help us continue our momentum, ask us questions, and watch our videos on our new YouTube channel. This will be edited and uploaded shortly. And thank you everyone for joining, and we'll see you next month. Be on the lookout for our next updates. Thank you, Dr. David Rawlings, for joining us.
Thanks for all the great questions.
Yeah.